This power stroke is the myosin head snapping back. And when it does that, it releases the byproducts of that hydrolysis reaction, adenine diphosphate and the phosphate it broke off. Okay? So we don't necessarily care that it releases those things. That's fine. It broke them down, used the energy to cock the head bound to the active site, and then the power stroke was it snapping back, going from a high energy state to a low energy state. Okay? The mousetrap, snapping. Okay? This is its low energy state. It took energy to cock it and load it. So oftentimes in the body puts the energy into setting everything up, like setting up a bunch of dominoes or whatever for it to all fall down nice and easily. Okay? The resting membrane potentials. We had the sodium-potassium pumps using energy to pump the sodium out and the potassium in, so that when we open the channels, they just rushed in passively through diffusion. Okay? So with it bound to the active site, it's going to pull the thin filament over the top of the thick. Now, the myosin head's still in that active site of the actin. So what happens next is another ATP molecule comes and binds to it. That causes it to let go. The myosin ATPase still on that head hydrolyzes that ATP, cocking it again. It sticks its head in another active site and repeats the process. Okay? So think of all those myosin heads. Well, half of them are attached, the other half aren't. And they just randomly are grabbing active sites and sliding them down until that process is shut down, until we go into kind of relaxation. Okay? So it's not like one of those Viking rowboats where they're all rowing in unison, because if we did that, there'd be a certain point where all those oars are out of the water, all the myosin heads are out of the active site. That wouldn't be good. So it's more of a random, alternating type thing. So another ATP molecule binds to the myosin head, causing it to release. We hydrolyze that ATP molecule, cocking the head again so it can extend further down, grab another active site on the thin filament. Half the heads are bound to the thin filament, while the other half aren't. This kind of prevents slippage back to the way things were. And this is how the sarcomere shortened. The Z discs come closer, but the actual myosin, pardon me, and actin filaments didn't change shape, didn't change length. They just slide over the top of each other. Okay. Yeah, we know that already. Come on, let's go a little further. Oh, bless you. So there's that calcium being rained down on there. We exposed the active site. So they're going to show that here, I think. Come on. Shape change. There's the active sites. The myosin heads are going to bind. Please. There we go. And then when they snap back, we call that the power stroke. Okay, it's going to release the ADP and the phosphate. Now, what cuts it to let go here? What's going to come in? Another ATP, good. And so basically the process just keeps repeating itself. There's the ATP, causes it to let go. Myosin ATPase hydrolyzes it, cocks the head again. Now we're speeding it up. Oh, I guess we weren't. Okay, well, that's what we got. Frustrating. All right, cool. So we need to be able to stop this. So we just stop all the little sequences along the way. So if we stop the nerve impulse, we stop releasing acetylcholine, right? If we don't release acetylcholine, then we don't have a local potential or an end-plate potential. So then we can't have an action potential. So we're not sending those signals down the sarcolemma, down the T-tubules. If those stop, then we're not going to allow the calcium out of the sarcoplasmic reticulum. Calcium's not going to be bound to troponin and tropomyosin. Okay? Or if we remove the calcium that was there, the thing will shape change back and block the active site. So now there's nothing for the myosin heads to bind to. So we don't worry about the muscle then contracting. Right? So we just basically stop everything we've done. So the relaxation phase, steps 14 through like 17, we end the nerve stimulation. We don't release the acetylcholine, and now acetylcholine esterase actually breaks down the acetylcholine that was there. And that ends our stimulation to the muscle cell. All right? Now, the calcium that was released is actually going to get pumped back into the sarcoplasmic reticulum. And it's saying it uses active transport. When something uses active transport, that tells us what? Is that a passive process, or does that require energy? Requires energy. So that's going to require ATP to set this thing up again to work when we want it to. It's going to pump the calcium, because the other reason you know it's active, anytime you take an ion and you put it in a higher concentration, it's going to build up, build up, build up. That's against diffusion, right? So you have to pump. You have to require energy. You have to push it in there. Okay? So it binds to something called calsequestrin. Think of a sequestered jury. It sequesters, it holds the calcium in the sarcoplasmic reticulum until we open up those channels. So ATP is actually used in muscle relaxation as well. If it's pumped back into the sarcoplasmic reticulum, then it can't bind to the troponin, tropomyosin, so it shifts back, covers the active site. This tension production maintenance stops, and the muscle fiber returns to its resting length. All right. How do you guys feel about that? Those steps. There's a lot of steps, but you feel kind of like you got an idea what's going on there? Because if you kind of got an idea, it doesn't take as much to kind of put it together. If you're just memorizing like these phrases and whatnot, that's harder. If you kind of have a visual or an understanding or this is what's going to happen next, then it's much easier. How many of you think you're going to see that on the test in an essay form? There we go. Like, all right. So at the end of the semester, when you're like, you really should tell us the essay questions. Really? You didn't know? Come on now. Does anybody have any questions on that? I mean, I know it's going to take you some time to look at, but is there anything that's confusing to you? Sarcoplasmic reticulum. Okay. And so with essays, it's just bullet form. You don't have to write an essay or a paragraph. I don't care about that. Just a bullet form of what's going on. And it really is, if you can get yourself going, oftentimes you can think about what's going to happen next and what's going to happen next and what's going to happen next and what's going to happen next. Okay? No questions? We feel pretty good? All right, let's talk through it one more time real quick without the pictures up there. This whole thing starts with a stimulus from the neuron, right? That comes down to the end of the neuron that we know is called the knob. She called it the bulb, well, not quite right, but she meant knob. And she was trying, so I appreciate that. So the synaptic knob, the motor end plate, terminal portion of the nerve, all those things mean the end. And it opened up what? Channels, good. What went through the channels? Calcium. What was the stimulus to open them? A voltage change. So they were voltage-gated calcium channels. Okay? So even if you couldn't quite remember, you kind of worked through it a little bit. Calcium comes in, causes what gets released? Acetylcholine that was being held in the synaptic vesicles through the process of exocytosis, good, and we release it across that space where the synaptic knob doesn't quite touch the sarcolemma. We call that the synaptic cleft. It moves across there and it's looking to bind receptors, okay, on the junctional folds, on the sarcolemma. It binds those receptors and it opens up now channels. What would we call these channels? Chemically gated. Who comes in? The salty guys, sodium channels, all right? We call that our end plate potential or local potential. If it's strong enough, well, if it hits something then with a T threshold, then it was strong enough that it caused an action potential. Good. The action potential gets propagated or conducted down the sarcolemma, then down the T-tubules, opens up what, where? You had calcium channels in the sarcoplasmic reticulum that responded to the voltage change, so they were voltage-gated calcium channels. Calcium released binds to troponin. The troponin, tropomyosin, changes shape, exposing the active sites. Good. Now, myosin head, ATP is going to bind to the myosin head, and what kind of little reaction are we going to have? It's called a, well, you were getting there, you jumped just a little ahead of me. It's called a hydrolysis reaction, which myosin ATP has hydrolyzed the ATP, cocking the head. The head then binds to the active site, and then we get the power stroke. Okay. Power stroke snaps back, pulls the thin filament over the thick, right? What causes the head to let go? Another ATP binds to it, it drops out, hydrolyzes that ATP, loads it, high energy state, binds to another active site, snaps back. And that keeps happening until we get to the relaxation component where we stop nerve stimulation. We break down the acetylcholine in the synaptic cleft with acetylcholine esterase. We pump the calcium that's been released back into the sarcoplasmic reticulum to be held by calsequestrin. The active sites are now covered up by the troponin tropomyosin complex, and our contraction stops, right? That's what I think. I'm going to do good on the test. Oh, yeah. Yeah. Kind of like what's in there explaining it. I forget who I've talked to. Like, my 201 class is here. My 202 is in the next room over. They look exactly the same when I'm standing in here. Labs look the same. So it's like Groundhog Day for me. Sometimes I don't know which class I'm in. I know it doesn't always seem that way, but all I'm looking for is you showing me that you know something. For instance, if you were on a practical and you called me over and you said, oh, hey, are you wanting me to say Selaturcica or are you wanting sphenoid bone? At that point, if that's what it is, I know you know it. So you know what I'd probably do? I just tell you what the frickin' answer is, right? Write it down. You got it. So if you come up here, if you're not sure, or if you don't want to come up here and ask, write down what you know. As long as it's right, and you're giving me even more information than I need, all I'm looking for is you to demonstrate that you know it. Then I can give you a point. You know, in an essay question, oh, they have that on there, good. They have this on there, good. That's all you see little check marks. I'm saying, oh, they knew this, they knew this, they knew this. That's what I'm looking for. I just can't give you credit for things that you don't show me that you know, if that makes sense. I can't assume that you know it, okay? And sometimes, I can tell you, I'm not speaking to any one person, sometimes on answers, you guys that maybe didn't understand it, but you memorized a line, and maybe there were 9 words in a bullet from the notes, and you got like 7 out of the 9 words on there, but you added something or whatever that made the statement incorrect. I can't give you credit for that. And I've had you come in my office and say, oh, but it's so close to what was in the notes. Yeah, but if that's what you think, then you're wrong. I can't give you points for that. So that's why the understanding is right, because it's not about the words that are in the notes. You may have 5 different words, it explains it just as well. That's all I'm looking for. Oh, hey, yeah, you got it. You know, okay? All right, so here we go. We got a question. What is the result of acetylcholine esterase being inhibited in some way? So we now know how things work, and this is one of those like critical thinking type things where I haven't specifically told you this, but you have all the information you need to figure it out, as long as you know what flaccid and spastic and neither mean. So what do we think would result if acetylcholine esterase was inhibited? Which one? Flaccid? Okay, he's going with flaccid. So what's flaccid mean? Limp, all right? Limp. Spastic means contracted and locked up. Okay, so what does acetylcholine esterase do? It broke down the acetylcholine. Okay, so if we don't break down the acetylcholine, it stays in the receptors. What does the muscle not ever do? Relax. So it would be paralyzed, but it wouldn't be flaccid. It'd be spastic. It'd lock up. It would seize up. It wouldn't have the ability, lose the ability to relax. This is what, when you hear of some wacky country somewhere using like nerve gas on their own people, that's what most of those nerve gases do. They'll inhibit acetylcholine esterase and cause them to go into a spastic paralysis. They'll shut down their diaphragm and they'll suffocate and die. And they'll die in this like convulsive type thing as their muscles seize up on them. If you ever had like a muscle do that on you, it's not very comfortable. Okay. Yeah. Botox doesn't do that. Kind of does the opposite. It does more of a flaccid paralysis. It paralyzes the muscle so it's relaxed, so it doesn't pull on the skin, and then you don't have the wrinkle. But then you lose the ability for some of those facial muscles to change your facial expression. So if people have had a lot of Botox, it's almost like they have a mask on. They can't change their facial expression. Okay? And I don't know who celebrities have or haven't, but sometimes politicians or whatever, you see some of those people, they always look the same. It's kind of weird. It kind of freaks me out. Like you guys unintentionally kind of freak me out because your facial expressions, I'm looking out there like, are they getting it or are they not? And it's just... So it's weird. I don't know how to take it. It's like back when I was dating. I just assumed they were into me, but I wasn't getting those signs. Until they came home with me. All right. Just kidding. A little bit on a very smaller scale at a localized level, yeah. But so she's asking like the leg cramps when you get like a Charlie horse. That's a little bit of what those folks would go through. And this, it's just all over their body. Okay. The leg cramps aren't from a problem with acetylcholine esterase. It's usually a sodium-potassium issue just locally at that point. They're not hydrated or they don't have enough potassium in their system. So then they drink Gatorade. Gatorade fixes all. All right, so now we're moving into types of contractions. So obviously, that was a big component of this unit, how muscles contract. Okay, so obviously it's going to be a big point question at the end of the test. Obviously, no brainer there. So isometric versus isotonic contractions. First one up is an isometric muscle contraction. In an isometric contraction, tension develops, but the muscle doesn't shorten. It doesn't change length. All right. So they're kind of showing you, or if somebody handed you, if I was standing up here with my arms like this and you handed me something heavy, okay, the muscle would have to contract so that I didn't drop it, but the muscle isn't really shortening. I'm not moving it. I'm just holding it, okay? That's an isometric one. Those of you that have ever played basketball or volleyball or whatever, when they have you do wall sits, those fun things that coaches do because they're sadistic, all right? Your muscles are contracting, but it's an isometric movement. You're really not moving anything in the joint, okay? Isotonic muscle contractions are more of what we think of. The muscle contracts and it shortens, okay? there's actually two phases to that. There's the concentric contraction. Tension develops while the muscle shortens. And tension can develop while the muscle lengthens, the other way. So sometimes weightlifters will call that the negative, okay? So you're bench pressing. The concentric portion is you're pushing the muscle up. Your triceps, your pectoralis are contracting and shortening and pushing the weight up. As you're bringing the weight down, the muscle's still contracting because you're lowering it nice and controlled, but the muscle's being stretched or lengthened, okay? Oftentimes, the eccentric contraction is more important. Will build muscle faster, okay? This is going to probably predate a lot of you. Cape Fear, an older movie with Robert De Niro. Okay, it's a pretty good one. So once again, when you don't have time, when you're no longer studying, watch there's a newer version, probably wasn't very good, but Robert De Niro played this crazy person, blah, blah, blah. But in that movie, he's pretty jacked up. And it was my understanding, I read something that he did like a 6-week eccentric training thing to get bigger and whatever for that role. And that's strictly the muscle lengthening while you contract. Now, athletes do that stuff today. Box jumps, jumping off a table and catching your weight, okay? My legs have to contract, but the muscles are actually being stretched as I land and kind of cushion that jump. And then they'll add to that. Not only will you drop down into that, then you'll jump up from there. And that'll help build your vertical jump or speed. It's training for more explosive type maneuvers versus just strength, okay? Because that's the other thing, I don't think we touch on it. How you train the muscle is how the muscle is going to function, okay? It's not all about just strength. Sometimes you're wanting to train it for fast contractions, explosive contractions. Because if it was all about strength, then those offensive linemen that can squat 600 pounds would be able to jump the highest. And we know that's not the case, right? Or run the fastest. All right, so any questions? Why might you have somebody do an isometric contraction instead of an isotonic one in some field that one of you might go into? Well, the difference with the isometric is there's no movement taking place. So if you've had a knee surgery, oftentimes they start you off with doing nothing other than just tightening the quads in your knee. You're not flexing the knee's straight, but they'll have you tighten it up and hold it and relax. Because if there's no movement, then you're protecting the joint, okay? But you're still working the muscles. You're not working them as hard as you could, but you're working them, which is better than nothing. Does that make sense? So if you're going into physical therapy, there's a time and place for some of the isometric contractions first, and then you go into some of the other ones, okay? The other thing with the eccentric, you're always stronger in the eccentric than the concentric, okay? So if I'm, once again, any of you guys, if you're bench pressing, all right, and let's say you bench press 225, whatever it is, that's what you normally work out with. That's cool. And let's say that's your max. Let's say you could do 1 or 2 reps. If you only could do that, pushing up, pushing down, you could probably take 300 pounds and lower it down. Okay, right? Slowly. Then you just need somebody to get that damn thing off your chest. So you'd have to have a spotter for that. Okay? I always choose to get a big spotter because when I'm done, I'm done. Get that shit off me. Looks like a Beavis and Butthead skit. Is there anything worse than getting hung up with like something and working out like that? You guys haven't experienced that humiliation? Oh my God. Where your muscles just basically say that's it, and you're pushing, and the weight's coming down, and you're like, hey, need a little help over here. Somebody's just staring at themselves, and you're like, no, dude, get over here. I'm dying. And because when you're done, you're done. Like, there's nothing left. It's like, get this thing off of me. All right, so now we get into muscle twitch. All right. Muscle twitch, we're going to have this threshold. It's a different type of threshold, but threshold nonetheless. Basically a minimum that we have to have hit to get a reaction. And for muscle twitch, it's the minimum voltage needed to produce action potentials. So when they do twitch, oftentimes this is an experiment they'll do in lab. Did any of you in high school do the frog gastroc experiment? You piff a frog, you basically run something through its brain so it's brain dead, but everything else is still working. No. PETA doesn't let you harm frogs anymore, I bet. All right, well, then we won't even get into that. But anyway, it was an experiment you do. And you'd hook up some electrodes and you'd zap the frog and you'd cause its muscles to contract. You'd measure how high you turned it up. Okay. See what was the minimum voltage it took to get a response. That's a little bit about what we're talking about here. So another term we have is a twitch. A twitch is a quick cycle of contraction and relaxation lasting less than 1/10 of a second, produced by a stimulus at threshold. One single stimulus at threshold. So essentially, it's what's that minimum voltage that you had to shock that muscle with to get an action potential. Okay? This single twitch is not enough to do any useful work. It's not enough to get anything done from that frog gastroc or anything, because it's the smallest, like, little blip. The phases of this twitch are really simple. First of all, there's a latent period. It's about a 2 millisecond delay. This is where tension builds up, because anytime before you start to contract, the muscle builds tension, okay? It's almost like taking the slack out, and then you start to move the weight, all right? So this is this latent period where only internal tension but no visible contraction or shortening takes place. The elastic components that tighten is being stretched. So when the myosin heads bind and they start to pull the thin filaments over the thick, the first thing, the movement could take place at either end. It's going to basically take the slack out of that tighten. When the tighten can't slide any further, then it's going to pull the thin over the top of the thick. That's this latent period. It also takes a little bit of time. Part of the latent period is the calcium being released, binding to the troponin, shape changing. That takes a little bit of time. Now, we're only talking 2 milliseconds, so that's really small, but it can be measured. Then we get into the contraction phase where the external tension develops. We actually start to see the muscle shorten. And then always the end is the relaxation phase, the loss of tension. Muscle returns to its resting length. With that, we have the calcium returning to the sarcoplasmic reticulum where it's held there by what? Calsequestrin. Okay. So, we're going to basically talk about how that kind of experiment that you guys never got to do kind of works. So, this all has to do with a simple concept. If you increase the intensity of the stimulus, well, actually, I'm not going to even tell you, I'm going to ask you, do you think if I had some device and I hook it up to a muscle, and I can control how intensely we stimulate it, that as I increase the stimulation, the contraction strength is going to go up? Look at that. So they do that experiment, kill those poor frogs, to teach you something that I could tell you in 5 seconds, and you're like, oh, that makes sense. So that's what's going to happen. What they're showing you in this picture, here's a nerve axon. Now, we haven't gotten there yet, but nerve axons are built the same way that muscles are. Muscles had fibers bundled together into groups called fascicles. Fascicles are bundled together in what we call a muscle. A nerve has nerve fibers. We bundle them into groups called fascicles. We bundle the fascicles into things that we think of as nerves running through the body. So down here's the stimulus intensity. So what voltage we hit it with. We hit it with just a little bit. It wasn't enough to stimulate any of the nerve fibers. Okay? So we up the intensity a little bit. We zap it again. Still wasn't strong enough. The third time we zapped it, we actually hit what our threshold was. This is how we determined the threshold. We didn't know this little red line was here until we hit it. It stimulated one of the nerves and got a little bit of a contraction because this recruited a very small motor unit. Contraction, relaxation. Okay? That's our twitch. So we want to see what happens. We hit it a little bit more. Now this recruited 2 fibers. Okay? And if we do that, then of course our strength of contraction is going to go up a little bit higher. All right, so we do it again. Now we got 4. We got a stronger contraction. Hit it again, higher. We get more. Contraction goes up. Hit it again here with number 7. We recruit all the fibers. We get our maximum contraction, but we don't know that just yet. Because what do? We hit it higher. But we see that it doesn't go any higher. So that we know that at a certain point, we max out on all the recruitment we can get, right? And that's what happens in that experiment. You got that little kid that's stoned on smoking or sniffing the rubber cement. And he's just turning that shit up really high until the damn frog's probably on fire. It's getting hit so hard. All right? But you see that the contraction that they measure doesn't go up. All right? And so recruitment intensity, stimulus intensity, as the stimulation goes up, the tension goes up to a point. There's a maximum. All right? And that's what all this whole thing is going to tell us. So stimulating the whole nerve with higher and higher voltages produces a stronger contraction. This is because the more you stimulate it, the more motor units you're going to recruit. The more motor units you recruit, the stronger the contraction. We call this multiple motor unit summation. Summation means what? Adding something together. So we're multiple motor units. We're adding motor units together. If we add motor units together, we're going to have a stronger contraction. This is a way, and I'll get this stimulate. That's doing a little different, but it can, because it can cause the muscle to contract a little bit. So it's a little bit like that's not why they're doing it. They're, in a way, what they're doing is they're stimulating it, and they're wearing it out so that when they take it off, the muscle has to relax. They've kind of worn out the muscle. Okay, so this is called multiple motor unit summation. And the brain, in a way, works just like this by recruiting multiple motor units to do something that requires more strength, like lifting. I think it's going to come up and say a gallon of milk versus a glass of milk. Okay, so this is one of the ways the body, the brain, controls the force of muscle contraction in a muscle by recruiting more or less or fewer motor units as it deems necessary. Did somebody else have a question? I didn't. I'm going to guess that probably they could get some muscle contraction from them as long as it was a nerve. Well, I shouldn't say that. That's going to recruit them. If they were paralyzed because the nerves was severed somewhere, okay? If they stimulated above the sever, no, because it's not going to cross. If they stimulated something below, they could probably get some sort of twitching. Not enough to, like they could be controlled necessarily, but yes, I would guess you could. So we just need somebody that's paralyzed. We'll bring them in here and we'll get one of the officers to tase them and we'll see what happens. That'll be our new experiment. We'll get in trouble for that. We could put it off as it's science and we're learning something from that. So here's some things that can happen when they do these experiments. Okay, this one, they're not focusing on stimulus intensity. Okay, they're going to keep that constant. All right, they're going to pick an intensity below the maximum, but above the threshold. Something in there that should get a recruitment of a motor unit. Instead, what they're going to look at here is the frequency. Okay, frequency means how fast they hit that muscle with that stimulus. So here you can see that this is a slower frequency. And I'm making this up. They don't label the numbers. Let's say this is a second. Each one of these bars is a second. So they're hitting it every second. We get a contraction and a relaxation, a contraction and relaxation, a contraction and relaxation. Here, they did the bars differently, but now they hit it faster than that. Okay? And they're going to tell us eventually it's going to be less than seconds if we're going to be talking milliseconds. But we can see what happens when they increase the frequency. The force of contraction went, which way to go? Up. Okay? So this means that not only if I increase the intensity, I increase the force of contraction. If I increase the frequency, I'm also increasing the what? Yeah, I'm increasing the force of contraction, right? Yes, thank you. Good God. Something so I feel like I can move on. So what happens, this is a good question. What happens is, part of the thing that regulates how strong it contracts is the amount of calcium. So what happens is you hit it before all the calcium got pumped back in. Okay? So once again, I'm making up numbers just to illustrate a point. Well, let's say with each stimulation we release 100 units of calcium. This only got to pump 50 of them back. So there were 50 already there. The next stimulation comes and we release 100 again. Now we've got 150. Contraction went up. Only 50 got pumped back, so now there's 100 left over and we hit it again. Now there's 200. Contraction went up. Okay? So that's what's happening. And we call these, it has names for it. But here in this low frequency stimulation, so this is considered low frequency. Each one of these bars is about 1/10 of a millisecond. So up to 10 stimuli per second, so 1 every 1/10 of a second, is considered low frequency. Each stimulus produces an identical twitch response. It contracts, it comes all the way down and relaxes. Okay. If we increase the frequency to what they consider to be moderate frequency, 10 to 20 stimuli per second, each twitch has time to recover. Now we see that it recovers because all of the red lines come back down to the baseline. So we're looking at this. This is the recovery. But we develop more tension each time we do it. Okay? So with each subsequent stimulation, tension increased, which we see because this went up and up and up and up here. This is called TREPI, or sometimes called staircase phenomenon. And it's a result of all the calcium not being completely pumped back into the sarcoplasmic reticulum. Okay. So low frequency, moderate frequency, high frequency. I'm waiting to make sure everybody's got everything written down they want. So high frequency is next. This is now 20 to 40 stimuli per second. Okay. Now, this rarely ever happens in the body. Okay. Greater than 25 stimuli per second almost never happens. But we're also going to continue to see that gradual increase in strength. But what's not happening now, looking at this graph? It's not having a chance to recover or relax. So now, this is what you're going to start to look like in a spastic paralysis. Okay? The muscle can't relax. Each stimuli arrives before the last one recovers. They piggyback on each other. This can be called temporal or wave summation. And what results is a sustained fluttering contraction, a spastic paralysis type situation that we sometimes call incomplete tetanus. So the contraction looks kind of choppy, like they're trying to relax or trying to go back to normal, but they can't, or they can't do it very well. In lockjaw? Okay. Probably. So that sustained contraction would be the same. It's not probably from increased frequency of stimulation, but yeah, because of some breakdown and it's inhibiting the acetylcholinesterase or something, yeah. So the results would be the same. The path you got there would be a little different. High frequency, we could go to max frequency. Here you saw, here's the high frequency. You went up high enough, the muscle would not relax at all. You would be in a full-on complete tetanus is what they're going to call it. Muscle has no time to relax. The twitch just fuses into one long contraction. We call it complete tetanus. This doesn't really happen in the body. Now, once again, not the same thing from frequency, but there used to be a Gatorade commercial. It was a distance marathon runner that was doing a Gatorade commercial. And the commercial, you might have seen it, they show him kind of rounding this corner for the finish line that's maybe 100 yards away, and his legs just kind of seize up on him. He looks like all of a sudden he's got like muscular dystrophy, and he kind of and falls down, and he can't get back up to finish. That's a kind of an incomplete tetanus that he's going through. Not from this, but that's what it would kind of look like. But like I said, now he drinks Gatorade that keeps everything good, and he can't be stopped. Okay. Any questions on that stuff? All right. Wow, we're moving right along. We'll be test time again before we know it. So now we're going to look at how we get energy for muscle contractions. Okay. The sources of what's our energy currency in the body? ATP. So how do we generate this ATP to do what we're going to do? And here are some of these types of things. So all muscle contractions depend on ATP. So how do we produce the ATP that we need? Okay. So normally, we're almost out of time. We've got 5 minutes. Do I want to go down this path? I don't think there's any reason to maybe start that. All right, we'll stop it there.